Hello everyone. So I'm here to talk today to encourage all young women to get into the STEM. So I'm going to talk a little bit about how I got here and what my research is about. So my name is Laura and I'm originally from Spain, but I'm here in Montreal doing a Ph.D. in neuroscience at McGill. Moving from Spain to Montreal was both challenging and exciting. It meant leaving behind family, friends, and a familiar way of life. But it also opened up a world of new opportunities that I know I didn't want to. That I knew I wanted to take full advantage of. So this is me as a kid, maybe not thinking about becoming a neuroscientist yet, but I was definitely curious about how everything worked. I was the type of kid who always asked why and tried to find an explanation for everything. The curiosity about the world around me never went away, and little did I know it would eventually lead me to a career in neuroscience. Fast forward a few years, and that curiosity led me to where I am today, exploring how the brain works in ways I never imagined back then. So I actually started my life in neuroscience while I was still in high school. So you're never too young to start. I did my final project of high school at a lab studying the composition of crab sticks. So certainly not neuroscience, but it was my first step into the scientific process. I learned how to think critically, analyze data, and how to solve problems in real time. And that experience led me to realize that I wanted to pursue research further. So the supervisor I had at the moment recommended me to another lab where I did an internship for the first 2 years of my undergrad. There, I was able to understand what it really means to be a scientist. It's not just about asking questions. It's also about how to find the answers even when the path isn't clear. After that, I spent 2 other years to do my final project on the undergrad at another lab. And thanks to all this experience, I was able to be accepted at another lab to do my master's, where I studied Alzheimer's. My passion for Alzheimer's wasn't just scientific, but also personal. Have you ever experienced something so personal that it made you want to change the world around you? For me, that was my grandfather. He had Alzheimer's, and watching him struggling with memory loss was both heartbreaking and motivating. I saw that this disease affected not just him, but my whole family, and that sparked something in me. I wanted to understand what was happening in his brain, and how I could help others facing the same challenges. So that's what led me to study Alzheimer's for my master's. In my last year of undergrad, I did an exchange in Montreal. I loved the city, and I decided I wanted to come back. And during my undergrad, I also realized I loved neuroscience. So I started looking for diverse universities to do my PhD in. And I realized McGill was one of the top ones. So it was the perfect opportunity to come back to Montreal while pursuing my dream of doing research in a top university. While I was looking for supervisors, at the beginning, the only thing I knew is that I wanted to work in learning and memory. And although I was first focused more on Alzheimer's, my focus has been shifted to autism spectrum disorder, or ASD. For my PhD work in Dr. Nachum Sonnenberg lab, that actually now has more women than men in the neuroscience part, I'm searching for metformin, which is a drug that is used for diabetes, to repurpose it to treat ASD. So I'm looking at the mechanisms, how it affects ASD behaviors, and also learning and memory. So now that I've talked a little bit of how I got here, I want you to give a little bit introduction of what I'm doing in my current research. So the first you need to know is what autism spectrum disorder, or ASD, is. ASD is a neurodevelopmental disorder that affects millions of people around the world. Despite that, we still don't fully understand all the mechanisms, and there's no cure. So that's why research like mine is so important, to be able to find ways to find treatments for these people. Instead of giving you a full list of all the features that people with ASD have, I wanted to show you a video of how it would feel to live with ASD.
This is the story of a boy who didn't talk for a long time. The boy liked things to always be the same. Any changes would scare and upset him. The unknown was an unfriendly place. The boy was very sensitive to lights and sounds. So he built secret hiding places where they couldn't get in. The boy didn't like looking people in the eye. He wasn't trying to be mean, it just made him feel uncomfortable. Sometimes he would flap his arms again and again.
One day, I found out I have something called autism. My family got me help. Slowly, I found my voice and learned all the ways I could live with it better.
Early intervention can make a lifetime of difference. Learn the signs at autismspeaks.org.
So you can see in this video that boy had autism, and as it says in the video, it is really important to find a treatment that can help them. And if it's an early intervention, it's much better. That's why I'm trying to find a treatment that can begin to be administered early in life. So now that you know a little bit more about autism, do you know a way that we can study it without directly studying humans? So one way is to use mouse models. So one question that I get all the time when I explain my research is, can mice get ASD? The answer is yes, they can. Or at least we can generate mouse models that mimic the symptoms of ASD. So as you may know, our body contains chromosomes that hold DNA that encodes for genes. These ones give the proteins that our body needs. And excess or annihilation of these proteins can result in diseases. What I'm doing in my project is that I'm using a mouse model that is called a knockout mouse model. So it lacks a specific protein that it has been related with ASD. So it's a perfect mouse for this disorder. Specifically, I'm working with the SHAN3 and the FMR1 knockout mouse models. They are related with two syndromes that present ASD. The first syndrome, which is produced by a deletion of the SHAN3 gene, is called Phelan-McDermid syndrome, or PMS. People with this syndrome present low muscle tone or hypotonia, which affects their ability to move and hold their posture. And they also show delayed development in speech and intellectual disability. All these symptoms make it harder for them to interact with the world around them. The second syndrome is the Fragile X syndrome, which is produced by a silencing of the FMR1 gene. Like PMS, people with this syndrome present delayed development in speech and intellectual disability, but they also have seizures and behavioral problems like anxiety. So some of you may have heard about metformin. So it's a common drug for diabetes, but what is exciting is that a lot of drugs that has been created for one purpose can be used to treat another disorder completely different. Think of metformin like bubble wrap. Did you know that it was originally created as a wallpaper? And then some people discovered that it was perfect for another thing, like protect fragile things. So like bubble wrap, metformin was originally created for diabetes, but I'm studying if it could help people with ASD. Okay, one thing that I love, aside from science, is cooking. And I found that this is a really good synergy with science. So for both of them, you need to follow a recipe and have all the ingredients and the material ready to go. So the first thing that you need is a hypothesis. In my case, that metformin is a treatment for ASD. Then, to prove this hypothesis, we need the mice, the metformin, and the material for all the experiments. So, in order to see if metformin could be a possible treatment for people with ASD, we first have to test it in mouse models. These mice, because of their genetic modifications, have behavior and symptoms that mimic the ones seen in humans with ASD. And here I'm studying how metformin affects their learning and memory and the ASD-like behaviors. Also, we look at how it works in the brain, see the specific proteins and pathways affected by metformin that could explain why we see a decrease in the ASD symptoms. To do so, we need 4 groups of mice. First, we need the mouse without the disease, that is the wild-type mouse, and then the mouse without the disease, that is the knockout mouse. And we're going to have them with and without treatment. I know that a lot of people here are not in these walls, so to make it simpler, we're going to call them Will and Kevin. So first, we're going to talk about the learning and memory test. So we did 2 different tests, the novel object location and the novel object recognition. So for the novel object location, what we do is that we put the mouse in an empty cage for 10 minutes and we let them get used to it. Then on the next 2 days, we put 2 identical objects and we let the mouse explore them for 10 minutes. And then on the last day, we move the object to a new location and we expect the mouse to explore more the object in the new location if it has learned. And then for the novel object recognition, it's quite similar, but it affects other brain areas than the novel object location. And what we do here is that instead of changing the location of the object, we replace the object for a complete new one. Now let's have a look at the results. So here we have the graph that represents the object preference. That is the time that they spend exploring the new object or location compared to the total exploration time. So now we're comparing with that doesn't have the disease with Kevin, the knockout, that have the disease. It's fine. So we can see that Kevin, who has the disease, doesn't differentiate between the object in the old location and the new location. As we can see here, the preference is similar to 0. So that means that they have an impairment in this type of memory. But when we give metformin to Kevin, this preference index increases, meaning that metformin has rescued this impairment. Interestingly, when we look at the novel object recognition, we see that Kevin doesn't present any type of impairment in this type of memory. Finally, I want to show you an example of an ASD behavior. There is grooming, which relates with repetitive behavior. So what we do here is that we put the mouse in an empty cage for 20 minutes. The first 10 minutes is just for it to get used to the cage, and then on the last 10 minutes, we analyze how much time it spends roaming and how often it starts roaming. So when we see the results, we can see that Kevin have an increase in both the time spent roaming and the number of roaming bouts. And this is reduced with metformin treatment, meaning that metformin also rescues repetitive behavior. Okay, so we have shown that metformin shows promising results in improving the ASD-like behavior and learning and memory, which can lead to an improvement in the quality of lives of people with ASD and their families. And thinking that metformin is just our purpose drug is even more exciting. So I would never imagine that I would be where I am today. So I want you all to encourage you to see all the possibilities that science and technology have to offer, and see that they have the power to change life, not just yours, but from the people all around you. Thank you.